Have you ever stopped to think about how we actually make some of our most advanced medicines? It's not always about mixing chemicals in a beaker. We've actually figured out how to use living cells as these tiny microscopic factories that can build incredibly complex, life-saving proteins for us. So let's dive into this amazing world of cellular engineering. I mean, think about it. Insulin for diabetes, vaccines that protect us from things like hepatitis B, even those powerful antibody therapies that are used to fight cancer. These aren't simple little molecules. They are ridiculously complex proteins. So how on earth do we make them on a huge scale? Well, the answer is we've learned how to hijack the machinery of life itself. But it wasn't easy. The very first big challenge scientists ran into was something we can call the protein authenticity problem. You see, you can't just take a human gene, pop it into any old cell, and hope for the best. For a protein to actually work in the human body and be safe, it has to be a perfect one-to-one copy of the natural one. Okay, so your first thought might be, let's use bacteria like E. coli. I mean, they grow super fast and they're pretty easy to work with. But there's a huge catch. They're prokaryotic cells, and our cells are eukaryotic. It's a fundamental difference. They just don't have the right equipment to do all the crucial finishing touches that human proteins need. So a lot of the time, the final product they spit out is totally inactive. Plus, there's always a risk of getting nasty bacterial toxins mixed in. We needed a better factory. So, what are these finishing touches I keep talking about? The fancy term is post-translational modifications. But think of it like this. Building a protein is like assembling a high-tech car. The first step makes the basic chain of amino acids. That's kind of like the pile of parts. But then it needs to be folded just right, have certain bits clipped off, and get special chemical additions, the whole finishing department, before it can actually function. And this is a perfect example. This is how our own bodies make insulin. It starts out as this long, floppy, inactive chain called preproinsulin. Then, some very specific enzymes come in and act like molecular scissors, snipping off those extra pieces. What's left are the A and B chains, which then fold together to become the active insulin molecule we need to control our blood sugar. Bacteria, they just don't have the right tools for this kind of precision work. Okay, so if bacteria are off the table, where did scientists look next? Well, they turned to a cell that's a lot more like ours, a eukaryote, but still really simple to grow, the humble baker's yeast. And you know what? Yeast turned out to be a brilliant choice. We know its genetics inside and out, it grows like crazy on really cheap nutrients, and, this is the important part, it has the cellular machinery to do a lot of those essential finishing touches. Plus, it doesn't secrete a lot of its own proteins, which makes it way easier to purify the one we actually want. And best of all, we've been using it for bread and beer forever, so the FDA already recognized it as safe. And the success was incredible. This is just a small sample of all the medicines made using yeast factories. We're talking the hepatitis B vaccine, insulin, human growth factor. The list goes on. Yeast really became the workhorse for the entire biotech industry. But there was a catch. It wasn't perfect. Just when we thought we had the perfect little factory, a new problem popped up. It was a really subtle but absolutely critical detail that the yeast finishing department was getting wrong. It all came down to the sugar coating. This sugar coating process has a scientific name, glycosylation, and it's a huge deal. About half of all human proteins have these complex sugar chains attached to them. And it's not just for decoration. This sugar coating is vital. It helps the protein fold the right way, it protects it from getting destroyed too quickly in our bodies, and sometimes it even acts like a key that lets the protein talk to other cells. And here's the twist in our story. While yeast does add sugars, it follows a completely different recipe from human cells. It gets the sugar coating wrong. This slide shows the problem perfectly. Okay, on the right, in path C, you can see the elegant, complex sugar structure that a human cell would build. Now look to the left, at path B. That's what yeast does. It just keeps piling on one type of sugar called mannose, making these really long branching chains. It's called hypermannosylation, and our immune systems can spot it as foreign and potentially trigger a really dangerous allergic reaction. So it was clear we needed a factory that could follow the human sugar recipe, step by step. And this is where some of the most amazing genetic engineering comes into play. Scientists decided, okay, instead of just using a factory, we're going to build a better one. To do this, they found a new hero,
a different species of yeast called Pichia pastoris. They took this really promising cell and basically gave it a genetic superhero upgrade. You see, Pichia had a key advantage. Its natural sugar coating was a lot less crazy than baker's yeast, which made it a much, much better starting point for a complete overhaul. And the plan they came up with was just ingenious. First, they used genetic scissors to go in and delete the yeast gene that starts that whole incorrect sugar piling process. Step 2, they added a gene from another fungus that acts like a tiny gardener, trimming the sugar chain down to just the right size. Third, they started adding the human genes that add the correct human sugars in the right order. And finally, they installed the entire multi-part system needed to put on the final critical sugar cap, called sialic acid. And the result? Just look at path E on this slide. It's a perfect match. I mean, it is identical to the human version. They had successfully reprogrammed the yeast's entire finishing department. They had created a humanized yeast, a microscopic factory that could produce authentic, properly sugar-coated human proteins, ready and safe for medicine.
And what's so cool is that this incredible breakthrough wasn't just a one-off trick. It completely opened the door to a new way of thinking about these cellular factories, not as these fixed things, but as totally customizable toolkits.
Modern genetic engineering relies on these circular pieces of DNA called vectors to program the cells. And today, they're designed to be completely modular. Seriously, think of it like building with Legos. You have different bricks for different jobs. A brick to snap the DNA into the yeast's chromosome, another brick to tell the cell how to copy the DNA, a brick that acts as an on switch for your gene, and so on. And here's the truly wild part. These Lego bricks can come from totally different organisms. As you can see here, you might take an integration part from one species of yeast, a replication part from regular baker's yeast, a selection part from Pichia, and a really powerful on switch from a completely different yeast. You can literally mix and match the best parts from all across nature to build the absolute perfect, custom-designed factory for whatever protein you're trying to make. This quote really just sums it all up perfectly. It's not about finding one single perfect system anymore. It's about building this incredible, ever-expanding toolbox. This modular approach lets scientists create and fine-tune new factories for new medical challenges faster than ever before. So we've gone from just borrowing nature's factories to literally building our own highly customized, high-performance production lines at a microscopic scale. It just makes you wonder, right? With a toolkit this powerful, one that can build the most complex molecules of life with absolute precision, what are we going to build next?